Hello, everyone, and welcome back to PS Platypus. Today, we're going to be having a look at some physiology. So, last week we talked a bit about fertilization and how the sperm meets the egg. Today, we're going to be moving past that, talking about everything that occurs afterwards. So, talking about placentation, fetal growth, um, birthing process, and then puberty. So, all that will be covered in this slide set. So, it will be quite dense content-wise. But again, if you do have any questions, let us know. Otherwise, we'll be trying to keep it very simple and to the point so that it's very high yield for you and for your exams. So here is an overview of everything we're going to be covering in this slide set. So placentation and fetal growth with all the four subtopics there, the birthing process, and then puberty. Again, any questions, let us know. So we'll start off where we left last time. So we talked about fertilization in our previous lectures. In this lecture, we're going to be starting off where we left. So we have the sperm meeting the egg, fertilization occurring. And important to know is that fertilization most commonly occurs in the ampulla of the fallopian tubes. And then from there, we have it forming the zygote, which is got 2n number of chromosomes. And then from here, it's all about mitosis, really. So cell division. The zygote undergoes constant cell division and mitosis. It migrates along the fallopian tubes towards the uterus. And then by about day 3, it's reached what we call the morula stage. So it's got about 32 cells at this stage. And then it keeps on dividing, and it forms the blastocyst by day 5 to 6. Also, another thing to keep in mind is that it's constantly being nourished by glycogen while it's traveling along the way as well. But when it becomes that blastocyst, it stops from becoming this uniform distribution of cells and it becomes polar. In other words, there's two distinct ends to it. There's one end where the inner cell mass is being pushed to one side. So you can have a look on the diagram and see the top part of the blastocyst is where the inner cell mass is collecting. And then the other side is the other pole. So the inner embryoblast cells, inner cell mass, they're going to go ahead and become the embryo and the embryonic membranes. Whereas the trophoblast cells that line that blastocyst, they're actually going to go ahead and form the placenta. So some important things to keep in mind at this stage. Next, we're going to move onto the actual implantation process where this blastocyst actually implants in the uterine wall. So the blastocyst will implant into the endometrium, and then a couple of things will happen. So the trophoblast cells, which we talked about before, which is that outer layer of cells, they actually, again, further divide into an inner layer and outer layer, which can be kind of confusing because we talked about the inner cell layer before, which was, if we go back, is the inner embryoblast. That's separate to the inner and outer layers of the trophoblast. So the inner layer of trophoblast cells, that outside covering, becomes what's called a cytotrophoblast. And essentially, it just divides and gives you more and more trophoblast cells. Whereas the outer layer, which is called the syncytiotrophoblasts, will actually merge their membranes and do something very important later on. But first, quickly, you would have seen the word syncytio come up quite a lot, both in the context of sperm and also in the context of RSV, or respiratory syncytial virus. And in both those contexts, it refers to how through some process, it causes membranes to merge. And it's very much the same here. What's happening here is that the membranes of these cells are merging and they're forming a larger mass that's secreting proteolytic enzymes that allow it to invade and digest the endometrium. So that's what's allowing the blastocyst itself to be able to create a gap or cavity for itself to fill in. And so it creates these lacunae or gaps. And that's got two functions. One is that when you're digesting the endometrium, you're going to yield nutrients, which is good for the placenta or good for the implanting blastocyst. But also, when you create those gaps can actually fill up with blood from spiral arteries. So it's also nourishing the blastocyst in another way as well. And at this stage, you also start secreting the hormone beta-HCG, which, as you know, is very important because it forms the basis of the pregnancy test. And it also allows the pregnancy to continue as one of its main functions. So clinically, you're going to be covering a couple of pathologies at this stage. One important thing to understand is that implantation has a pretty high failure rate with about 2/3 of blastocysts failing to implant and 1/3 of implanted embryos actually being miscarried. So the next thing clinically to learn about is ectopic pregnancies. So we talked about how implantation normally occurs inside the uterus in the endometrium. What can actually happen is that the blastocyst can actually sometimes erroneously end up implanting outside the uterus within the fallopian tubes themselves. And as you can imagine, there's not much space or the right conditions for placenta or an embryo to grow in that area. And so you're going to have an ectopic pregnancy, which is actually a medical emergency. And you'll learn more about that later on as well. And then finally, we have placenta praevia, which is a bit of a strange condition. I recommend looking at an image of it online. It's essentially where the blastocyst will implant near the cervical opening. And so the placenta actually covers the internal vaginal opening. And so during labor, rather than it being the baby or the neonate being delivered first and then the placenta coming out, because the placenta is actually on that cervical opening, it will be delivered first before the baby is born. So another interesting condition to have a look at. And again, images of it will make it clear why exactly the structure and orientation causes that to occur as well. Next up, we have the formation of the placenta. So we're now going to talk about how exactly we go from an implanted blastocyst within the endometrium to forming this placenta, which is the interface between the maternal and fetal circulations. So what happens is the cytotrophoblast cells we talked about before, which again are formed by the inner layer of trophoblasts, they're going to expand into the lacunae and gaps that were created before by the syncytial trophoblasts and the digestive enzymes. And when they do that, they initially form what's called the primary chorionic villi. And these are sort of anchoring structures that anchor the embryo or the developing fetus to the endometrium. So they form an initial anchoring structure, but then what happens is the mesoderm of the blastocyst, it actually penetrates through the primary chorionic villi and it forms these secondary chorionic villi. So we get a secondary structure coming out of it. And you can have a look at the diagram as well and see how the structure of the villi slowly changes as the mesoderm invades through it. And then finally, probably like the most important form is the tertiary chorionic villi, where we have fetal blood vessels going through and invading through those chorionic villi. And that is what allows there to be a link between the fetal and maternal circulations to the placenta. And there's two diagrams showing that, the top and the bottom one. And the bottom one also shows a bit of a cross section, so you can see how the structure of those villi changes as it gets invaded by mesoderm first and then by blood vessels. All right. And now we come to the structure of the placenta itself. So I think it helps to have an idea about where the placenta is located relative to the developing fetus. And you can have a look at the diagrams as well that show kind of where it sits in the abdomen, in the uterus. And so in terms of structure, we consider it to be sort of a fetal component, which is where the fetal blood supply is coming in, and the maternal component. And they should be pretty easy to understand given that we've covered how exactly the placenta has formed. So the chorionic villi that we talked about before, the tertiary chorionic villi, where the fetal blood vessels invade into the secondary mesoderm villi, that is the fetal component, the chorionic villi. The maternal component comes from the spiral arteries that, if you remember, were filling up gaps or lacunae created by the trophoblast cells that were digesting the endometrium. So again, there's two components and they're sort of explained there as well. And then the function of the placenta is to provide nutrients and remove waste. So it's nourishing the developing fetus and removing any waste products that it's creating. It also actually makes a couple of hormones, and you can have a look at those hormones in the graph down below. So you can see there's beta-HCG, there's progesterone, there's estrogens. And an important thing to keep in mind as well is that during most of pregnancy, the progesterone is actually higher than the estrogen, which is very important because progesterone plays a very important role in driving and also helping with some of the adaptations associated with pregnancy. The third point there is that this placenta is impermeable to large proteins, and which is really important because it serves as an immunological barrier. So it means that large proteins from the fetus won't enter the maternal circulation and then invoke an immune response where the maternal immune system will try and attack the fetal developing fetus. It also means that red blood cells, for example, won't cross over, so that if there's differences in red blood cell proteins, there won't be any immune response to that. And then the final thing here is that, however, it is still permeable to some noxious chemicals. So alcohol, for one, so that's why you have fetal alcohol syndrome. Nicotine can cross over. Certain drugs can cross over. Hence, you learn about certain drugs that are teratogenic or can cause complications during pregnancies. And also some pathogens as well can be vertically transmitted. For example, HIV is one. All right, so on the right, you can have a look at some of the images that explain that in more detail. But we're just going to move on to the next slide now and have a look at some of the extra embryonic membranes. So you've already covered this a little bit in terms of embryology in previous lectures, because you had a look at an image like this during GIT embryology. But just to revise a little bit, the chorion is this outermost membrane that forms the placenta. So you can see it sort of merges with and forms a placenta down below. The amnion, however, is a transparent sac with amniotic fluid, which you would have learned quite a bit about in medicine. And it's got a couple of important roles. The first one is to nourish the actual developing fetus, but also to protect it. So it acts sort of like a cushioning fluid that protects the fetus inside from trauma. And then we've also got a couple of other functions that are sort of minor, but it actually gives space for the fetus to flex and exercise with its growing muscles. It contains sort of the fetal urine, the lung liquid, nasal secretion, skin cells. That's all sort of recycled and gotten rid of later on. A couple of things to keep in mind here clinically, though, when it comes to the amniotic fluid, is oligo and polyhydramnios. And that's where you have under or overproduction of that amniotic fluid. Underproduction, you can imagine, is undernourishment and low protection, whereas overproduction can actually cause excessive pressures that actually squeeze on the developing fetus and can cause certain effects from that as well, which you'll learn about later on. Again, it's assessed by ultrasonography, but at this stage, all you really need to know about it. The yolk sac is a structure whose importance decreases in size and it decreases in size and importance during early development. But you, again, will learn about it a little bit during GIT embryology. It actually plays more of an important role in the development of other species. So it's sort of a vestigial structure almost in humans. And then the allantoid is a small outpocketing that forms the base of the umbilical cord. And you can have a look at it on the diagram there, that green structure there. All right, we'll now move on to the next section, which is about the maternal adaptations that occur during fetal growth and fetal development. So the first one is an obvious one, it's weight gain. That's because you have the developing fetus, but you also have all those membranes that are developing the placenta. You've got all that fluid as well, filling up in all those sacs. So you're gonna have some natural weight gain associated with that. But you also have some other adaptations besides that as well. So with the cardiovascular system, there'll be an increase in extracellular fluid. That way you have more fluid that you can pump along and supply to the fetus. And then you also have increased cardiac output because you're supporting the metabolic demands of both the mother and the developing fetus. You've also got a decrease in total peripheral resistance that's driven by progesterone, and that just will allow for diffusion more readily. Increased red blood cells, again, that's to deliver more oxygen to meet the higher metabolic demands of both the mother and the developing fetus. Importantly, there is an increase in red blood cells, but it actually leads to a mild anemia because even though we have more number of red blood cells, you actually have a lower overall concentration because of the higher increase in the extracellular fluid. So you're actually going to have a mild anemia here. Respiratory adaptations include increased ventilation. So you're just breathing in more to be able to meet the oxygen demands of the fetus. But as a result of breathing excessively, you're also going to be having a lot of CO2 being blown off as well. So you're going to end up with a slight respiratory alkalosis because a lot of CO2 is being exhaled. A decrease in residual volume as well, because if you imagine the developing fetus as like a large abdominal structure, it's going to limit the diaphragm's movement, and so it's going to limit how much you can inhale and keep in your lungs, and also limit the amount that you can that's remaining in your lungs at the end of expiration as well, because of that obstruction there. Metabolically, a common number that's quoted is that the caloric intake of the mother is split with a third to the mother, a third to the placenta itself. So that receives quite a lot, and then a third to the developing fetus. You also have insulin changes that occur to be able to increase the blood sugar levels, to be able to meet the metabolic demands of the developing fetus. And then finally, in terms of your renal system, because you have an increase in cardiac output from the heart, you're also going to have an increase in glomerular filtration rate. but you're also going to have more fluid retention. Even though you're filtering out more, you're actually going to have more fluid retention because the aim is to retain a lot of plasma to be able to exchange substances with the fetus. And then you have GIT changes. You have a increase in motility actually rather than a decrease. Decrease in motility of the GIT system. And that's because if you imagine the food is being made to go through more slowly in the GI tract, and that way you're absorbing more and more nutrients from it for the fetus. So you might get a bit of constipation, but that's because your body's trying to absorb as much as it can from the food that's being given to it for the developing fetus. And then finally, your other important change is your joints become more flexible. And that's because of a hormone called relaxin. And that allows your ligaments and muscles to relax a little bit so that it can make the changes necessary to be able to accommodate the growing fetus. All right. And now we come to birth. So we're going to talk about this in a couple of stages. The first thing we're going to talk about is uterotrophic and tonic factors. So during development of the fetus, you might remember we talked about how there's a high level of progesterone that's present during pregnancy. And that progesterone's high levels is what makes the myometrium, or the muscular component of the endometrium, be sort of quiet, relaxed, and not really doing much. And also keeps the cervical canal closed so that there's no changes that would cause birthing to happen early. But just before birth, the level or the ratio of estrogen to progesterone gets much higher, so the estrogen dominates. And that's what drives the birthing process. So you're going to have uterotrophic factors and uterotonic factors. And we'll talk about what those are in a second as well. The uterotrophic factors, if you imagine what the word means, uterus trophic environment, it's creating the environment or the preparations that you need for the uterus to be able to contract. So for example, estrogen would be a chemical that does that. It does it in a couple of ways. So it stimulates contractile proteins, oxytocin, prostaglandin receptors, gap junctions in the myometrial cells. And so it causes hyperplasia and hypertrophy of the myometrial cells. So an increase in the number and an increase in size. And the uterotonic factors are, again, uterotonic. So uterus, tonic is actually the muscle itself and the contractions of the muscle. So it's going to induce uterine contractions in this case. So oxytocin and prostaglandins like PGF2 alpha and PGE2 are examples of hormones that do in fact induce uterine contractions. And clinically it's important because let's say someone's not going through labor and you want to induce labor, you can actually inject these chemicals to induce uterine contractions. So they work in a variety of different ways to cause the uterine contractions. But another important thing to also be aware of is the Ferguson reflex. And that's sort of a positive feedback loop where what happens is the baby moves further inferiorly into the birth canal during the birthing process. And in doing so, it presses on mechanical pressure receptors in the cervix. And that sends excitations up to the hypothalamus. And that signals to the posterior pituitary to produce oxytocin. And then the uterus contracts more vigorously, and that causes the head to go out further. And then it creates a positive feedback loop where until it keeps on occurring until the birthing process is complete and the baby is completely out. So it peaks during that second stage of labor, which we'll see in a second. And now we come to the stages of labor. So we're talking about second stage of labor being the baby delivery stage, but we'll start from the very beginning. So stage 1 is cervical dilation. And that's where you have contractions onset. So when we have the contractions starting to the full dilation of the cervix, that's the sort of time period it occurs in. And at this stage, you have regular and short contractions followed by a period of relaxation. And the muscle factors of the uterine fundus gradually shorten over this period because of the heavy contractions that they're undergoing. And you can have a look on the diagram there as well as to where we are in this stage. And then stage 2 is the actual baby delivery stage. So moving on from the dilation to delivery of the neonate. And that's, we talked about before, the whole reflex that occurs to be able to allow that to happen, the Ferguson reflex. And then finally, once the baby is delivered, the placenta is still left inside. So we have to deliver that as well. And so stage 3 is finally the expulsion of the placenta. And remember, we talked about placenta praevi, which is that medical condition where the cervix or the blastocyst implants near the cervical opening. So the placenta is actually delivered first because it covers the cervical opening and is delivered first. and then the baby is delivered after. So that would be where 3 comes before 2. But just have an overview of what the labor stages are and what roughly occurs in each of them as well. And now we come to lactation. So moving on from birthing process to breastfeeding. So we'll talk a bit about breast tissue development and how it actually forms. So breast tissue is derived from skin tissue or the ectoderm, which sort of makes sense looking at where it is. So it's ectodermal in nature. And then at birth, it's only present as a primitive lactiferous duct. And so they're not very functional at that stage. And then it's at puberty, during exposure to estrogen, the ducts of the breast undergo rapid growth and branching, and there's also formation of rudimentary or very basic lobules, but again, still not very functional. And then during pregnancy, there's rapid lobulo-alveolar growth. So the lobules and the alveoli, they rapidly grow, and then you start having some basic secretions. So you have internal secretions of immunoglobulin, but nothing is really happening just yet because the high levels of progesterone and estrogen are inhibiting prolactin from really doing much or making the secretions actually occurring. And then finally, after birth, so during the lactation stage, full milk secretion is possible. And that's because prolactin and oxytocin is sort of peaked. It's able to do what it does after birth, and we have galactopoiesis or maintained milk production. Yeah, so that's the breast tissue development. And then we come to lactation stages. So there's 2 main stages of lactation, so 1 and 2. 1 is the time from mid-pregnancy to 2 days after birth. And during this stage, we have our glands differentiating, and they're still secreting small amounts of casein and lactose, which are referred to as the colostrum. But there's a high level of progesterone that's actually restricting the milk production. So if you had a look before as well, we said that the oxytocin is not able to actually induce, or prolactin is not able to induce milk secretion because the high level of progesterone is inhibiting it. And then during lactogenesis 2, we have secretion of copious amounts of milk because the progesterone sort of dies away, and prolactin is able to do what it does. And you have the milk letdown reflex, which is when the baby sucks on the nipple, mechanoreceptors activate oxytocin from the posterior pituitary and prolactin from the anterior pituitary. And as a result, the oxytocin will induce milk ejection, so the actual letdown of the milk now for the feeding baby, whereas the prolactin from the anterior pituitary is going to cause milk secretion that prepares milk additionally for future feedings as well. So have an idea of how that works roughly as well. And then we come to puberty. So puberty, we only have a couple of slides on this because there's not too much in terms of content, but it generally refers to the period of time between childhood to adolescence. And when we talk about puberty, you'll normally talk about the growth spurt as well that occurs during puberty. And this growth spurt is described as having 3 main stages. There's the takeoff, which is the time of maximum growth. Then there's the PHV, or time of peak height velocity. And then finally, epiphyseal fusion, where you have those growth plates fusing over and then growth sort of ceasing at that point in time in terms of height. And an important thing to keep in mind here is that boys start about 2 years later than girls when it comes to that takeoff and peak height velocity with their growth spurt, which might make you think that they'll end up having a shorter height. But because they are starting off at a higher baseline, they've had more time to grow for that baseline. At the end, on average, they tend to be taller at the takeoff period. And so overall, will end up being taller at the end as well, which is what the graph sort of demonstrates there. All right. And on the right there as well, you can have a look at some of the relevant hormonal pathways that, again, you'll learn about in more detail when it comes to particular pathologies, particular physiology as well. So the first one up there is the HPG axis, or hypothalamus, pituitary, and gonad axis. And you'll learn about that in the context of menstruation, and also in the context you would have learned of sperm and its production as well. And then finally, at the bottom there, you have what's called the HPA axis, or the hypothalamus pituitary adrenal axis. And it's important for reproductive system because if you remember, the adrenals are also important because hormones produced include androgens, and those androgens play an important role as like a sex hormone as well. And they're also implicated in a lot of pathologies like PCOS, which we'll learn about later on. All right. Now we come to female puberty. So when it comes to female puberty, the reason we cover it in particular is because there's a couple of really important terminology that we have to learn about that is relevant to this particular area. So the first one is adrenarche. So that's, like we said before, the HPA axis, the hypothalamus pituitary adrenal axis. Adrenarche marks the activation of the HPA axis. So a theme you'll see is that all of these terms describe sort of firsts that occur during the puberty development, and adrenarche is the first activation of the HPA axis. And so you start having your adrenal androgen secretion prepubertally. Pubarche marks the start of puberty, and it's the appearance of pubic hair. Gonadarche, as would make sense, is the activation of the HPG axis, and that's what sort of starts that growth spurt and gonad development. Telarche is the appearance of the breast tissue. Menarche is probably a very important one to know because you'll be using it both here, but also in the context of clinical skills when you're going to ask about menarche, because the timing of menarche and also the period of time between menarche and menopause, so the duration of the periods, is important and implicate in a lot of pathologies. And so you'll be asking about that in clinical skills as well. So it's important to know that menarche means the onset of menses. So it's the first period, and it's usually 2.6 years after puberty, although that age is coming down over time because one theory is that the more nutrition and the better conditions that we have, menstruation is able to occur at a much earlier age. And one reason for that is explained by the kisspeptin and leptin pathways. So the way it sort of is explained is that leptin is a hormone that's produced by or sort of coded for by the OB gene, but more importantly, it's produced by adipocytes. So it's produced when fat cells secrete it, and it'll reduce hunger and also increase the level of KISS1, which we'll talk about in a second. But if you imagine, if someone has a high level of adipocytes because they're very well nourished, they're going to produce a lot of leptin. So they're going to have low hunger, which makes sense. Like the more you eat, the less hungry you're going to get. But they're also going to have a higher level of KISS1. And that'll become important in a second, because KISS1 is responsible for increasing the levels of FSH and LH, and therefore causing the onset of puberty. So if you are well nourished, you have high levels of leptin, less hunger, high levels of KISS1, high levels of FSH and LH, and then earlier onset of puberty. However, even though you have an earlier onset of puberty, it's often described as being a slower puberty than it normally would be. So overnutrition causes that. But then if you have a complete absence of KISS1, you end up with hypogonadic tropic hypogonadism. So that's a problem with the level of the gonads, with the KISS1 being incorrectly transcribed and translated. And as a result, you have underdevelopment of the gonads. All right. And then we come to the Tanner stages of puberty. I didn't find this too high yield. There wasn't much to talk about it in terms of the OSCEs, like it doesn't really come up there. And even in the exams, you might have your odd multiple choice question about it here or there. But if you do want to go to the effort of remembering it, I know a table can be a bit hard to memorize. So one mnemonic that's sort of used online, and one that I used when I was learning this topic, is using your hand as a way of remembering different stages. So the way it's taught is 1, 2, 3, 4, 5 are the 5 stages on your fingers. You can remember 1 and 5 as being the start and the end. So the start being prepubertal, and then the end being at the adult stage once you've finished puberty, right? Where you've completely finished it, you're done. And the numbers that are coded for it for the adult stage is about 15 years old. Therefore, the rest of the fingers, your index finger, right, is going to be stage 2. And that you could sort of remember the age for that as well, because stage 2 in Roman numerals resembles the number 11. So stage 2 is around 11 years old, which is easier to remember once you have that mnemonic in mind. Let me close that. And then we come to stage 3, which is 13 years old. Easy to remember: 3, 13, 4, 14. So you've got the numbers there in terms of the stages, and also the associated ages as well. Then it comes to the actual developments that occur during those time periods. At stage 2, you typically have breast budding in females and testicular enlargement, and then you also have pubarchy, so pubic hairs appearing as well in males. And you can have a look at the table and see sort of correlates with that as well pretty nicely. And then stage 3, it's where you have your breast mound, your first breast mound appearing and enlarging, and also the penis lengthening. And that is sort of taught as your third finger is your longest finger. And so you can remember it as being the stage where the penis lengthens and also the breast mound starts to enlarge as well. So that's stage 3. Stage 4 is now where we have menses starting, right? So that's menarche. And then we also have both breast mounds now appearing and then enlarging. And then you also have your penis widening and the testes growing larger as well. That's the final stage of, sorry, the fourth stage, stage 4, which is at 14 years old. And so you can sort of try and remember it using the hand mnemonic or the table, whatever works for you. But that's sort of the end of the physiology slides. If you have any questions, concerns, feedback, or any queries about what we've covered today with the content, let us know in the comments. Otherwise, that's it for today's physiology lecture, and we'll see you in the next one.